Well, good morning, everyone. I'd like to speak about the coming wave of biosimilars and what they mean. Are they similar enough? So the idea of biosimilars is really started before 2009, but with the passing of a specific law by Congress, BPCIA, which is the Biologics Price Competition and Innovations Act. And that created a pathway for the FDA to develop and approve biosimilars that would be cheaper and more readily and more available than the original patented drugs. There are increasing numbers of biosimilars coming into the oncology market as patents expire, and we'll go through the list that's available today. I'd like to make everybody aware that biosimilars are not exactly generic medications. They are similar, but they're not identical. And really, we want to know how and do we know that these similar medications are as good as the reference or the original medication. So I'd like to give you a little case presentation. Mrs. Schwartz, a 53-year-old female, who presented earlier this year with anemia, and the workup showed multiple and diffuse liver metastases as well as lung metastases with thickening of the sigmoid colon, and that was biopsied and shown to be colon adenocarcinoma by colonoscopy. So with extensive stage 4 disease involving the liver and the lung, she was started on Folfox and Avastin, and received 4 cycles with good tolerance and response on CT scan. So now you are going into the EMR to sign her next chemotherapy orders, and you now note that instead of Avastin, bevacizumab, it says bevacizumab AWWB 5 milligrams per kilo. So is that good, or are you angry? And is it a good idea to sign this order? Take a moment to answer for yourself. So I hope by the end of this talk you'll understand better what the questions are. So what is bevacizumab AWWB? A generic, a biosimilar antibody, a plot perpetrated by insurance, or Mvasi from Amgen, that is a biosimilar of bevacizumab? Take a minute to answer. And the answer is specifically D, Mvasi. And what is this an example? Biosimilarity, interchangeability, or substitution? So as you'll see, what this is substitution, where the pharmacist has decided that instead of the brand name bevacizumab Avastin, they'll give you a biosimilar instead without you ordering it. So let's take a look to start out with the BPCIA of 2009. This is from the Congressional Record. And Title VII, Improving Access to Innovative Medical Therapies, Biologics Price Competition and Innovation Act. And the sense of the Senate was that biosimilar pathways should be established to balance innovation and consumer interests. And so those are the key issues with this pathway and this kind of medication. So as we can see, looking at the actual wording of the BPCIA of 2009, the biologic product is a biosimilar to a reference product based on data derived from analytic studies, animal studies, and a clinical study or studies. So those are the 3 key things. And to blow that up a little bit so you can see it, it's a biosimilar to a reference product based on data derived from analytic studies, animal studies, and a clinical study or studies, and pharmacokinetics and or pharmacodynamics that are sufficient to demonstrate safety, purity, and potency in 1 or more appropriate conditions. And furthermore, according to the law, the biologic product and reference product utilize the same mechanism or mechanisms of action, and the condition or conditions for use of the prescribed, recommended, or suggested in the labeling proposed for the biologic product have been previously approved for the reference product. So that's pretty detailed and seems pretty good work for Congress, especially looking back from 2020 point of view. But that has really created the pathway that the FDA uses today with those specific details of the law. So, it goes back a little further, including this Patient Protection and Affordable Care Act, signed by President Obama, creates an abbreviated approval pathway for biologic products that are demonstrated to be highly similar or interchangeable. So, what is a biosimilar? It is not the originally approved branded agent, which is called the originator compound. It's not exactly a generic because it's not a chemical copy, but it is a compound that's made by biologic cells, and it may be made in a different cell line or in different cell types altogether. But it is required to have the same amino acid sequence as the originator, but variable post-translation modifications may arise as it grows in these biologic cells. It should also be noted that the originator compound has a lot of variability also. So if you take the Avastin bevacizumab that was made in 2020, it's not very much like the original Avastin from 10 years ago. And in fact, the European and the American versions of Avastin are different too, because they're made in different plants. But the bands of variability for the different components and the chemical purity are quite small, and the biosimilars have to fall into the same bands. So again, by the FDA's rules, again, coming from the BPCIA, the biosimilar has to show that it is highly similar to the reference product, notwithstanding minor differences, and there are no clinically meaningful differences between the biologic product and the reference product. And then in order to make the higher standard of interchangeability, a sponsor must demonstrate that you can switch over and produce the same clinical results, even though you're alternating between the originator and the biosimilar. Like generic versions, biosimilars are intended to reduce prices by creating competition in the market space and a marketplace, and experts anticipate they have potential to reduce direct spending on biologic drugs by $44 million over 10 years. That is possibly true. I think there are questions about how the competition is going to happen now that they're actually more like branded generics due to the way the FDA has required they be labeled, but we'll see about that in a moment. So if we look at a generic versus a biosimilar, the size of the molecule is kind of like comparing my bicycle to an 18-wheeler, and a big one at that. And the complexity is a little bit like comparing my bicycle to the space shuttle. So these biosimilars are much larger and are much more complex than the generic type of molecules. So in a generic, you have a known and defined chemical structure. It's manufactured by chemical synthesis, and the molecular weight is usually less than 1,000 Daltons. And by any analytic method, these are identical chemicals. But for a biosimilar, it's based on cell manufacture. The molecular weight is greater than 150,000 Daltons, and there is a range of variability in terms of most of the chemical characteristics. So here's a little picture comparing aspirin to a monoclonal antibody. Aspirin, or acetyl salicylic acid, is 180 Daltons, and the antibody is about 150,000 Daltons. And that's the difference in size and, again, complexity between the generic molecule and a biosimilar. Again, generic drugs are copies of brand name drugs, but biosimilars, as we said, are highly similar and have no clinically meaningful difference, and they're basically brewed up in a biologic milieu. So to demonstrate biosimilarity, the pathway and some of the issues are, first of all, for a biosimilar manufacturer, there's no access to the reference product history, manufacturing process, controls, or parameters. So this is all proprietary information that the originator would hold on to. The originator molecule is a fully characterized reference product in terms of the structure, the higher order structure, so secondary and tertiary structure, receptor binding, stability, biologic function, and general properties. So these all have to be kind of duplicated by the biosimilar. All these have to be within a certain range, a band range. So you can see here their high and low tolerances, and then these parameters where you have a medium and then a variability, but they fall within the range of band. And then one would have to develop and identify a cell clone that meets the predefined margins, establish cell bank and manufacturing process, and then establish biosimilarity by these criteria that we have already discussed: analytic studies, non-clinical studies, comparative clinical pharmacology, pharmacokinetics and pharmacodynamics, and then clinical safety, efficacy, and immunogenicity in at least one clinical trial. So the biologic source of variability is basically because these are not manufactured or chemically synthesized, they are grown up in living cells. They are basically brewed, and glycosylation is variable in different cell lines and in different cells that may even be in the same cell line. So the glycosylation can lead to altered biological activity, altered PK, changes in antibody function, and most important is some of these things that can affect the antigen-dependent cytotoxicity and also immunogenicity. So you've got glycosylation, fucosylation, galactosylation, and so forth that can all have different biologic effects. So this is a picture of some of the processes that are used to make biologics. I was lucky enough to tour the Genentech plants. So this is kind of like a pilot plant where they first brew up 50-liter batches, and then they go to these larger 250 to 300-liter batches. And then these are the large Cefidex type columns that are used to purify the proteins. And in fact, it's not that different than brewing your favorite beer. Basically, it is an art and requires care and feeding of the cells. So the FDA approach to biosimilar approval looks like kind of an inverted pyramid compared to the originator development. In the originator, the analytic studies are kind of the smaller part, then the preclinical studies, then the clinical pharmacology, and finally, the clinical studies are the largest, most expensive, and most time-consuming. But for biosimilar development, it's been turned on its head. So a lot of time has to be spent on the analytic characterization of the structure and function, then preclinical activity showing that it binds to the target and actually can inhibit or whatever it does add to the target, and then clinical pharmacology, including PK/PD, and one clinical study in an approved situation. So extrapolation, that is necessary for biosimilar use and approval. So because of this inverted pyramid, there are very many fewer clinical studies, but the FDA would approve it based on the larger amount of in vitro and in vivo studies. And some of the scientific justification for evaluating are shown here. Is the mechanism going to be different? Do the pharmacokinetics and pharmacodynamics vary? Is the immunogenicity expected to vary? And are there differences in expected toxicity? So assuming that all these things can be extrapolated, then if you show activity in one indication, the FDA gives you approval for multiple indications. So the biosimilar only has to do one clinical study in one indication and then gets the approval for all indications despite one clinical trial. And again, they always talk about that extrapolation is based on the totality of evidence plus knowledge of the reference product plus scientific justification. So these are the ways that the FDA justifies the broad approval that it uses. So here, for example, are the package inserts for Avastin on your left and Mvasi, which is the Amgen biosimilar. And you'll see that they look pretty similar. The black box warnings are the same. The indications are the same, except for metastatic breast cancer, which was revoked. But you can see that even though they only did a study in colorectal cancer, they get approval for non-small cell cancer, glioblastoma, whatever, and the wording's exactly the same. So as far as the FDA is concerned, once they give you biosimilar status, you get to take all the indications of the originator compound. So interchangeability, let me just again show why that's different. That means you can switch off. So if you started biosimilar versus the reference, if you started the reference drug, can you give the biosimilar for a couple courses, switch back again, and at least 3 switches, and then show the same endpoint at the end? This is the FDA guidance and criterion for interchangeability. And as of June of this year, no oncology product has received interchangeability status for any biosimilar. This is not true of some of the rheumatologic drugs, however, which have done those switching studies, but in oncology, none of them have. So substitution is then a third issue. So substitution depends on the law in your state. So automatic substitution is a practice mandated by law where a pharmacist must dispense the less costly or preferred biologic regardless of whatever you prescribed and without prior approval of the prescriber. Substitution is when the pharmacist may dispense an alternative without the prior approval. And switching is a practice wherein a prescriber may change the prescription from one biologic to another. I think in our EMRs, we have a lot of switching going on, and it may or may not be with your approval. So here's a list of all the oncology biosimilars that have been approved as of July 2020. And there are 28 approved biosimilars today, 16 of which are oncologics. The first one was the biosimilar filgrastim, Zarzio. I'm sure many of you are familiar with it, in March 2015. So I've listed these actually by name of the originator molecule by brand name. So you see a couple of Avastins, one Epogen, quite a few Herceptins, a couple Neupogens, and then the most recent ones have been these rituximabs. And the manufacturer here varies from some companies that you're very familiar with, Merck that's working with Samsung, Pfizer, Amgen, Sandoz, Novartis has a whole biosimilar division, and so does Amgen. But you will be able to tell which these are if you go and look it up based on this last 4 letters that are the suffix. So the suffix is supposed to not make sense. It's not supposed to let you guess who the manufacturer is, but it is supposed to let regulators decide, track what the molecule is if there's an issue. The problem with this is that because these are so specifically identified now, every company has made them a branded biosimilar. So you see the 2 biosimilar Avastins are called Mvasi and Zirabev. So you can actually order them by name if you want, and similar for your Herceptins and so forth. And because they need to have this kind of suffix that specifically demonstrates them, all the companies are now giving them names. And so in the end, it's not quite as interchangeably biosimilar, I think, as Congress had imagined in the beginning. And that is as a due to the way that that the FDA has instituted these suffixes. So the non-proprietary names are specific to a defined active substance. So you would have bevacizumab, originator, biosimilar 1, 2, and 3. But they wanted to distinguish non-proprietary names in order to improve traceability, and therefore they came up with this kind of random 4-letter suffix to the non-proprietary name. And this has created kind of a way to identify each of the generics, but has landed up with branded biosimilars. So. Okay, so time for another question. So you're starting a patient on FOLFOX and bevacizumab for metastatic colon cancer. You open your order set in the EMR and you see the following options. What would you select? Avastin, bevacizumab reference, Emvasi, bevacizumab AWWB, or Zirabev, which is bevacizumab BVZR. So make a choice. So I think that actually in the end, we won't have a choice because our pharmacy committees and our purchasing agents are going to just decide on whatever is cheaper. And unless you specifically bring up the issue of interchangeability and switching, you will find that from time to time, whatever you're ordering may be a different biosimilar. It may not be the originator. And the question of should the patient be informed and should they consent to a use of a biosimilar instead of the originator is something you can discuss with your P&T committees, as I have with mine. So in conclusion, I just want you to remember that biosimilars are not exact copies or generics. They're made by living organisms and they are brewed. They come from a regulatory pathway enabled by the Biologic Price Competition and Innovation Act in 2009 by Congress, and then enacted by the FDA with their guidance in 2010, and further a part of the Affordable Care Act. The approval requires preponderance of preclinical information and 1 clinical trial. So contrary to standard drug development, they only need to show that it works about the same in 1 clinical trial. The manufacturing should be such that there's equal tolerability bands for all the chemical and binding assays and all the assays that are done of the reference product and the biosimilar. There are numerous oncologic and growth factors now approved. Interchangeability is not approved for any oncologic biosimilar today, and it's based on more complicated trials that require switching back and forth. The naming today requires the generic name or the non-proprietary name and a 4-letter suffix, and substitution should only be allowed if interchangeable and state laws allow. Again, you will be seeing these in your electronic medical records. You will be selecting them without really choice often, but if it's an issue, you should discuss it with your pharmacy and therapeutic committees at your institutions. So thank you, and I hope this has helped you kind of understand what's happening with biosimilars today in your institution and in an EMR near you.